Hi, my name is Konika Shallon, and I'm a practicing physician in Jacksonville, Florida, with two aesthetic and laser clinics. And I also serve as medical director and senior vice president of clinical operations at Candela Medical. It was interesting because they tracked very closely. What we did in the study is we used quantitative Visia imaging, and we looked at the brown spot counts on the side of the face that was treated versus the side that was untreated. And what tracked was that it's what we all see in clinical practice. The laser works very, very well from a cosmetic perspective. We can see that there's improvement when we resurface the skin in this fashion. And we saw that that improvement was very significantly different from the untreated side. So we had nearly 40% improvement in the treated side at the 1 month follow-up. And so when we looked at that and compared it to our epigenetic data, we found that it made perfect sense. That was indeed the time that we saw the most important changes for all of the visible type, aesthetic and cosmetic type of changes that we looked at. So it was very reassuring that what we see in clinical practice played out on a molecular level. But what was interesting is that beyond what we see in clinical practice, our body continues to respond 6 months out with some important changes that impact how our molecules are acting, how our cells are acting, and as they relate to aging and skin health. I think that this is really some of the most exciting information in that study. The study is very interesting in that it looked at all of our skin cells at the CPG sites, and it found that DNA methylation reversed so many important things at those CPG sites. And they fall into three different buckets. They fall into the cosmetic bucket, they fall into the aging bucket, but they also fall into the skin health or skin cancer biology bucket. And what we found is that the laser is modulating DNA methylation at very specific sites, such as FGFR3, HOXB4, UBE21, and so on. And those are all genes that are tied to keratinocyte differentiation and how the basal layer of our epidermis is regenerating. And that is important to the formation of basal and squamous cell carcinoma pathogenesis. So DNA methylation shifted these loci, and they moved in a direction that were associated with healthier cell regulation. In other words, less keratinocyte differentiation. And that really is important when you think about ways that we could potentially lower keratinocyte carcinoma risk. Now, it's also important to note that this is an early study, that this is a correlation, that we are looking forward to additional work that can prove causation. We haven't proved that this is skin cancer prevention, but what we do know is that our patients tend to have fewer basal and squamous cell cancers when we do certain types of energy-based treatments, specifically non-ablative fractional treatments like the 1940. And now we know that on the molecular level, we have proof that show that the genes that are associated with keratinocyte carcinoma formation are impacted. And those findings also result in kind of durable changes that also went out to 6 months for the whole duration of our study.